Hey guys, I'm going to go through some of the organelles from chapter 4. I'm going to talk about a few organelles here, the ones that are part of the endomembrane system. Endo meaning in, membrane, you know what a membrane is. So all of these organelles have a membrane and they are part of a system. They all work together. So as we're going through this, you need to make sure you understand what each of them do individually, but how they work together as a group. And think about, well, what if something went wrong at this point, what are the consequences of that? Some of the functions of this endomembrane system is protein synthesis, so making proteins, and we know a little bit about that. We've talked about it with the transcription and translation, starting in the nucleus and then moving out into the cytoplasm. So make sure you make connections about that and see some of the similarities as we go through this. Transport of proteins into membranes. So again, remember that membranes have proteins, you can see them in this picture, embedded into them with different functions. So this is how these get moved around. Also, the endomembrane system is part of carrying proteins out of the cell. So if the cell is doing the job making these proteins, it's going to have to get those to other parts of the organism, or if there's waste, it needs to get it out. So the endomembrane system functions in that, as well as making and moving around lipids. So remember, if they all have cell membranes are made out of lipids. So they have to be coming from somewhere. The endomembrane system is part of that, as well as detoxification of poisons, and you will see how that is in just a minute. So if you look up here in the picture, you can see all of these organelles are connected. So we have the nucleus, we have the endoplasmic reticulum, we have the Golgi apparatus, we have the plasma membrane right here, we have lysosomes over here, and it is a network, and you need all of them for the endomembrane system to be working properly so that the whole cell functions properly. One thing I wanted to highlight right here is that ribosomes are not part of the endomembrane system because they are not membrane-bound organelles. They are made out of RNA. So even though we see them show up, they're not part of this system. They just play a role in how it works. Excuse me. So first thing we're going to look at is the nucleus. So again, we're here. It's in the purple. We know that that's where the DNA is stored. In the middle of the nucleus is the nucleolus. In this region of the nucleus, this is where rRNA is synthesized, and rRNA is what makes ribosomes. Ribosomes are made out of rRNA. So that gets made there. We know that we learned about transcription, so you see right here, we're in the nucleus. You have your DNA. Enzymes come and they create a strand of mRNA that happens in the nucleus, and then it leaves through the structure of the nucleus, this nuclear pore, and it's able, and we've always said it's going out to the cytoplasm, so we'll see where another location for it to go is. If you look up here, you can see the nuclear pores, and that allows for the mRNA to leave, as well as any products made, like this rRNA, as well as some proteins that get made in the nucleolus. They need a way out, so it's through those nuclear pores. One thing I wanted you to look at in this picture is that when you're looking at the nucleus under a microscope, the DNA is going to look different at different points. When cells are about to divide, the DNA condenses, and that's when you see the chromosome shape. Otherwise, if the cell is not getting ready to divide, you just see like a blob of DNA. So be aware of that when you're looking at pictures. The next part of the endomembrane system we're going to look at is the endoplasmic reticulum. And pay attention to the order I'm going through these, because this is the order of the progression of the endomembrane system. So we have the rough ER and the smooth ER. If you look at this picture, it is consistent, it's connected, always connected to the nuclear envelope. And the purpose of that is when the mRNA leaves the nucleus, it's going to come out, and the reason why the endoplasmic reticulum, the rough ER, is rough is because it has ribosomes attached to it. And we know that ribosomes are where mRNA is translated into protein. So with these with these ribosomes attached to the outside of the rough ER, you're making the polypeptide chain into it's directly going into the inside, the lumen of the endoplasmic reticulum. And what's really cool about that is inside of the endoplasmic reticulum are specific enzymes that help the protein fold. And it also attaches carbohydrates to the outside of the protein. And these carbohydrates are like little identifiers so that the protein knows where to go. And once the carbohydrates are attached to the protein, it is now called a glycoprotein. Glycoprotein. Glycoprotein, again, is when there is a carbohydrate attached to the protein. And so once this protein is it's modified with that carbohydrate, it's now a glycoprotein, and we know that it's a secretory protein, meaning it needs to get out of the cell or go to the cell membrane. The ER buds off. So remember that the cell membrane is made out of the lipids, and so think about like a bubble, how like kind of bubbles can bud off of each other, like a lava lamp, or even like an oil drop. It buds off, and now we have a transport vesicle with the glycoprotein in it. So that was the rough ER I just talked about down here. The smooth ER is smooth because there's no ribosomes attached to it. So it has a different function. In the smooth ER, this is synthesis of lipids. So oils, steroids, and phospholipids are made there. Again, if we're synthesizing lipids, we're going through a chemical reaction. There has to be specific enzymes, and those are located inside of the smooth ER. It also is used to detoxify drugs and poisons. So what happens is there is also enzymes inside of the smooth ER that change the structure of if there's a poison inside of the body that gets into the cell. It makes it more hydrophilic so that it wants to leave the cell. It can leave the cell easier since this inside of the cell is water. And it also stores calcium. Once the glycoprotein, the transport vesicle, leaves the endoplasmic reticulum, it goes to the Golgi apparatus. And there's two sides of the Golgi apparatus, the cis and the trans ends. The transport vesicle enters through the cis end. There's different types of enzymes, again, inside of the Golgi apparatus. And remember, we're able to have these different environments inside of the organelles because they're membrane bound. And that's what makes them so special, is that they can do different functions, store different enzymes, and it's going to be different than what's in the cytoplasm. Inside of the Golgi, at different regions of the Golgi, there is different types of enzymes doing different jobs throughout. So once the the transport vesicle enters the cis phase, and remember, it's that made out of the membrane, so it's able to just kind of insert itself in, kind of, again, like oil, or like a raindrop, how when a raindrop hits another raindrop, it kind of becomes one big raindrop. That's what's happening here. Inside, once the proteins, the glycoprotein from the ER is inside of the Golgi apparatus alters those glycoproteins and the phospholipids, if that is a product from the ER. It alters it more, so it adds on more carbohydrates. And remember, those carbohydrates are for, it's like a marker, it's like an address. So if I draw a protein, it looks like this, and it has a specific carbohydrate marker on it, this marker can be detected, and it is going to be like an address of where it's going to go. So let's say that type of carbohydrate is bound for the cell membrane. Maybe a protein that needs to go to the mitochondria is going to have a carbohydrate with a different shape. And then once you get to the location where it's tagged, this shape is going to match up to a docking site at the location, and that's how it will know it's in the right spot. So then we're going through the Golgi. It is again being altered with these carbohydrates. Let's see what else. And tagging it for different parts of the cell. When it leaves through the trans side of the membrane in a vesicle that buds off again. And there will be inside of the vesicle, so if we're looking at a secretory vessel, there will be markers on the outside of this vesicle as well that will be recognized for the location that it needs to ultimately end up. So all in all, the Golgi apparatus, it packages, but it modifies and it sorts the materials from the ER, the proteins or the phospholipids that need to get to other places in the cell. Again, up here, I didn't mention this, but the Golgi also makes macromolecules. And then it will deliver those to other parts of the cell as well. So lysosomes. Lysosomes actually, they come from the Golgi apparatus. They bud off from the Golgi apparatus. And they have hydrolytic enzymes inside of them. They're digestive enzymes. So if there's anything in the cell that needs to be broken down, they will have specialized enzymes inside of the lysosome that will do that. For example, if there is a organelle that is malfunctioning, the lysosome will take up that organelle and it will break it down into its component parts, and it will release the carbon, hydrogen, oxygen, anything that organelle is made up of. It will release it back into the cytoplasm so that the cell can use it again. Anything on this that I need to talk about? No, so we're right here, lysosomes. So here you can see if food comes in, a food vacuole is created. It will combine with the lysosome. The lysosome has the digestive enzymes in it. It will break down the food and it will release the components of the macromolecules that the cell needs to use for whatever else. Vacuoles, this is mostly in plants and fungi, but they kind of work the same as a lysosome. So again, they'll have enzymes in it that digest food, but vacuoles also store organic molecules in plants. Sometimes they store poisonous molecules so that herbivores don't want to eat them. They also store their storage for inorganic ions for the plants. So you'll see those sometimes. They work very similarly to vacuoles a lot of times, or for storage. So I want to recap because you need to understand how proteins travel through a cell, because remember, proteins do all of the main functions inside of a cell. So we need to know where they're made, where they're going, how do they get to where they need to be so that the cell is functioning appropriately. Also, if there is other parts of the body or other cells that need something, how do they get it from a cell that is producing it? So let's walk through this. Again, we start in the nucleus, and this is where the mRNA is transcribed. The mRNA leaves the nucleus through a nuclear pore. It attaches to the ribosome on the outside of the rough endoplasmic reticulum. There, the protein is made inside of the endoplasmic reticulum, and it is modified into a glycoprotein. Oh my goodness. Glycoprotein, again, just a protein that has a carbohydrate attached, and that is for identification purposes. It leaves the rough endoplasmic reticulum, I'm right here, in a vesicle, which then combines with the cis side of the Golgi apparatus, where again it is modified further with more tags. It is packaged. The proteins in the Golgi apparatus are modified so that they have tags on them so they know where to go. They are put into a vesicle that buds off from the Golgi. That vesicle will then, if the target location is out of the cell, it will go to the cell membrane, will release the protein into the extracellular matrix. If there is a mess up in a protein, it can be taken up by a lysosome and digested, or the protein could be the enzymes, the digestive enzymes, which will turn into a lysosome. So remember these steps. There's one here. This is step 2, step 3, step 4. You have to make sure that you understand how a protein moves through the cell, and you have to understand that if you see that there's a connection, so you shouldn't be able to identify if there's something goes wrong, what else is that going to affect. All right, thanks guys.